Hi, in this module we will talk about soil health and plant nutrition management. Let us start with soil. What is soil? Soil is a thin layer of Earth's crust which serves as a natural medium for the growth of plants. Let us understand about the soil profile. Soil profile, as you can see on the screen, is a vertical section through a soil. It represents sequence of soil layers which are differentiated from each other. There are 5 horizons in soil. The topmost horizon is O, then comes A, then comes B, then C, and the last one is R. Let us study them one by one in detail. The first one, which is O, stands for organic. It is a very thin layer formed at the top of mineral soil resulting from decayed plant and animal residues in relatively undercomposed form. This horizon is commonly found in forest areas. The next horizon is A, which is surface horizon. It is also called A alluvial horizon. It is the surface layer of most organic matter accumulation and soil microbial activity. It is the uppermost layer and has alluviation of clay, iron, and aluminium oxides and organic compounds and other soluble constituents are found in this horizon. When alluviation is pronounced, a lighter colored E subsurface soil horizon is apparent at the base of A horizon. The third layer is B horizon, which is the subsoil. This layer accumulates minerals such as iron and aluminium oxides, silicate clay, and organic compounds. The fourth layer is C horizon, which is substratum. It is also called parent rock. It is the unconsolidated material underlying below the B horizon. It is considered as a zone of no major biological activity. The last layer is the R horizon, which is also called bedrock. This is the layer of partially weathered bedrock at the base of the soil profile. Unlike the above layers, our horizons largely comprise continuous masses of hard rock that cannot be excavated by hand. The next thing is components of soil. There are 4 major components of soil, and these are mineral matter, organic matter, water, and air. As we can see in the image, mineral matter is 45%, organic matter is 5%, and of that 5% organic matter, we have 80% humus, 10% root, and 10% organisms. Air and water are at 25% each. Let us firstly discuss the mineral matter in detail. It is generally composed of very fine broken rock fragments and minerals dominated by inorganic constituents or distinct minerals like quartz and feldspar. Primary minerals like quartz, biotite, muscovite, etc. dominate the coarser fraction of soil. Secondary minerals like silicate clay and hydrous oxide, clays of iron and aluminium, etc. are present in very fine fraction clay in soil. The next one we discuss is the organic matter. Soil organic matter contains partly decayed plant and animal residues, living and dead microbial cells, derivatives of microbial decay. Soil organic matter is the storehouse of nutrients in soil. It promotes healthy crops, supplies resources for microbes and other soil organisms, and regulates the supply of water, air, and nutrients to plants. Soil organic matter also reduces soil erosion, shades the soil, and keeps the soil cooler in very hot weather and warmer in winter. The third component of soil is soil water. Soil water dissolves the salt and makes up soil solution. This soil solution acts as an important medium for supply of different essential nutrient elements to the growing plants through roots. The movement and retention of water in the soil is primarily influenced by the characteristics of the soil, and these characteristics are soil structure, soil texture, nature of inorganic and organic colloids, exchangeable cation, pore space, etc. And the last component of soil is soil air. Air spaces in the soil consists of that portion of the soil volume which is not occupied by soil solids. Soil air influences the growth of the plants as well as the activity of the different microorganisms present in the soil. Soil air contains more carbon dioxide, very small amounts of oxygen and nitrogen than atmospheric air. Now, let us discuss soil health. Soil health is the fitness of a soil to support crop growth without deteriorating. It deals with the integration of important physical, chemical, and biological properties of soil. Soil physical properties include texture, structure, density, porosity, consistency, temperature, color, and water content. And soil chemical properties refers to measuring of soil nutrient carrying capacity such as cation exchange capacity, organic carbon concentration, and soil reaction. Soil biological properties refer to soil respiration, presence of earthworms, and microbial diversity. Microbial biodiversity. But what is a healthy soil? A healthy soil provides adequate levels of macro and micronutrients to plants and soil microbes. This reflects the ability of the soil to mineralize nutrients and a moderate pH, which is between 6.0 to 7.0, which allows the nutrients to be held in the soil and made available to the plants as needed. A healthy soil also has good tilth. This includes a good structure that resists degradation which is erosion and compaction. It provides adequate aeration and rapid water infiltration. A healthy soil also accepts, holds, and releases water to plants and groundwater. It promotes good root growth and maintains good biotic habitat that sustains high and diverse population of pests and pathogens. A healthy soil has low salinity levels and low levels of potentially toxic elements which can be boron, manganese, and aluminium. A healthy soil also has high resilience and is able to withstand deleterious events such as drought and flooding. Now that we have understood what a healthy soil is, let us talk about the sustainability. That is, how do we sustain a healthy soil? The sustainability of soil health and balanced usage of fertilizers for improving the crop productivity and profitability largely depends on the soil test-based nutrient management. But what is a soil test? Soil testing is an essential tool for knowing the soil capacity to supply plant nutrients in available forms to plants. Soil test refers to the chemical analysis of soil, and it is a scientific means of quick characterization of soil fertility status. What are the soil testing equipments? There are a variety of soil sampling equipments, and these equipments include khurpis, permanent marker pens, common stationary items like stapler, spade, soil sample information sheets or tags, polythene sheets or paper, and sampling bags. These all form soil sampling equipments. How do we take soil sample? There are 11 steps to it. First, we divide the field into similar soil types. These types are selected from the sampling points based on the visual observation and farmers' experience. So we need to divide the fields into similar soil types, slopes, erosions, etc. And then the sampling points are collected which are based on visual observation and farmers' experience. Second, we remove the surface litter at the sampling spot. Third, we make a V-shaped cut to a depth of 15 centimeter in the sampling spot using a spade. Fourth, we remove 1-inch thick slices of soil from the top to bottom of exposed V-shaped cut. Fifth, we collect at least 6 to 8 slices of soil samples from each sampling field. Sixth, soil slices collected from different spots should be placed on paper or a polythene sheet. Seventh, mix the soil thoroughly by removing plant roots and stones. Ninth, air dry the soil in shade at room temperature and put in cloth or polythene bag. Then tie the bag properly. 10th, we write the sample number on both sides of the bag with a permanent marker. And the 11th and the last step is to attach completely filled out soil sample information sheet to the soil bag and send the soil to soil testing facility. Soil sampling methods differ in orchards and polyhouses. When we are collecting the soil sample in an orchard, then we need to remember that fruit orchard requires 2 samples. One composite sample can be taken from under a tree, and one composite sample has to be taken between the trees. We then prepare a final composite by mixing the two. And when sampling is done in polyhouses, then we need to stop fertigation for 2 days. We then remove the upper 5 centimeter layer from these spots, make a V-shaped hole of 15 centimeter in depth between drippers, and then collect the soil. Let us look at the guidelines for sampling depth. When the crop is in grasslands, then the soil sampling depths in inches should be 2 inches or 5 centimeter when we are measuring the depth in centimeters. When the crop is shallow rooted crop like rice, groundnut, finger millet, pearl millet, small millets, etc., then the soil sampling depth should be 6 inches or 15 centimeter. When the crops are deep rooted like cotton, sugarcane, banana, tapioca, vegetables, etc., then the soil sampling depth should be 9 inches or 22 centimeter. And for the crops which are perennial crops, plantations and orchard crops, the soil sampling depth that we require would be 3 soil samples at 12 inches, 24 inches and 36 inches, or we still require 3 soil samples at 30 centimeter, 60 centimeter and 90 centimeter. Now that we have understood soil, importance of soil testing in maintaining soil health and how to do soil sampling, let us start with plant nutrition. The first thing is to understand what are the essential plant nutrients. An essential plant nutrient is an element which is so essential for specific metabolic activity that in its absence, plants cannot continue to grow or complete their life cycle, and this element cannot be substituted by another element. Essential plant nutrients makes the uptake of nutrient occur, and this occurs at both the root and the leaves. And secondly, plants can only uptake in organic form of nutrient, which is the mineral form of nutrient. Based on their relative requirement by plants, these elements are classified as macro and micronutrients. Macronutrients are further classified into primary and secondary nutrients. Primary nutrients are essential right from seed germination. They are hydrogen, oxygen, carbon, nitrogen, phosphorus, and potassium. Secondary nutrients are needed only when the plants have started growing or they have grown for some period. These nutrients are calcium, magnesium, and sulfur. Generally, plant contains hydrogen to the tune of 6%, oxygen to the tune of 45%, carbon also to the tune of 45%, nitrogen at 1.5%, phosphorus is at 0.2%, potassium at 1%, the secondary nutrient calcium at 0.5%, magnesium at 0.2%, and sulfur at 0.1%. The important essential micronutrients concentration in plants are chlorine, iron, boron, manganese, zinc, copper, molybdenum, and nickel. The composition of chlorine and iron is at 100 ppm. Boron, manganese, and zinc each is at 20 ppm. Copper is at 6 ppm. Molybdenum and nickel is each at 0.1 ppm. What is the role of nutrients that we just spoke about? Let us start with the macro primary nutrient, carbon. Carbon is used by green plants for photosynthesis. It is required for cell formation in plants. About 45% of plant tissues are made up of carbon. And carbon is also abundantly available in air. The next one is hydrogen. Essential for cell and tissue formation, hydrogen is obtained from water and it is required for energy reactions. Hydrogen forms about 6% of the plant tissues. The third macroprimary nutrient is oxygen. Oxygen is required for photosynthesis and respiratory activities. Oxygen helps in formation of tissues and cells. Plants take oxygen from air and water. Oxygen forms about 43% of the plant structure. The next one is nitrogen, an essential constituent of proteins, and it is present in many compounds of great physiological importance in plant metabolism. Nitrogen helps in harvesting solar energy through chlorophyll. Nitrogen imparts vigorous vegetative growth and dark green color to plants. It produces early growth and also results in delay in maturity of plants. Nitrogen helps in transfer of genetic information through nucleic acids. Now let us talk about phosphorus. Phosphorus gives rapid and vigor start to the plants and thereby it strengthens stem and therefore decreases lodging tendencies. Phosphorus brings about early maturity in cereals and counteracts the effects of excessive nitrogen. It stimulates early root development and growth and thereby helps in quick establishment of seedlings. Excessive phosphorus may cause the deficiency of zinc and iron. But phosphorus is closely related to cell division and development. And therefore, phosphorus is also important. The next nutrient is potassium. Potassium is an essential requisite for enzyme activation. Potassium provides the osmotic pull that draws water into the plant roots. Potassium imparts winter hardiness to legumes and other crops. It counteracts the harmful effects of excessive nitrogen. Potassium imparts increased vigor and disease resistance to plants. The next major nutrient is calcium. Calcium regulates the permeability of cell membrane. It is required by a number of enzymes for their proper functioning, which is lipase, phosphates, D, pound, amylase, and apirase. It makes the stem stiff and thereby reduces lodging in cereals. Calcium neutralizes the organic acid formed within the plant body and thereby eliminates their toxic effects. It accelerates nitrogen fixation in legumes and helps in increasing nitrogen uptake by plants. The next nutrient is magnesium. Magnesium helps in protein synthesis in plants. It is involved in photosynthesis and organic acid metabolism. Magnesium helps in synthesis of fats and increases oil content in oil seeds crops when it combines with sulfur. Being a mobile element, it is readily translocated to younger plants during its deficiency. And the next one is sulfur. Sulfur is required for synthesis of sulfur-containing amino acids, which are cysteine and methionine. Sulfur is needed for the synthesis of A-biotin, thiamine, or vitamin B1 and glutathione. It occurs in volatile compounds responsible for the characteristic taste and smell of plants in mustard and onion families. Sulfur also stimulates seed formation. Now let us talk about micronutrients. The first micronutrient is iron. Iron helps in formation of chlorophyll. It helps in absorption of other nutrient elements. A structural component of porphyrin molecules involved in oxidation, reduction in respiration, and photosynthesis. Iron helps in carrying out different enzymatic reactions in plants like cytochrome oxidase catalase, peroxidase, nitrogenase, etc. The next micronutrient is manganese. The role of manganese is closely associated with that of iron. Manganese supports the movement of iron in plants. It also takes part in oxidation reduction process and decarboxylation and hydrolysis reactions. Manganese is required for maximal activity of many enzymes reactions in citric acid cycle. Optimum manganese supply sometimes helps in countering the bad effect of poor aeration. The third micronutrient is copper. Copper forms various compounds with amino acids and proteins in plants. It helps in utilization of iron during chlorophyll synthesis. Lack of copper causes iron to accumulate in the nodes of plants. Copper also acts as electron carrier in enzymes, which brings about oxidation reduction reaction in plants. Next is zinc. Zinc influences the formation of some growth hormones in the plants. It is associated with water uptake and water relations in plants. Zinc also influences the activity of dehydrogenase enzymes such as pyridine nucleotide, glucose 6-phosphate, and triose phosphate, etc. Zinc also helps in stabilizing ribosomal fractions. The next micronutrient is boron. Boron acts as regulator of potassium to calcium ratio in plants. It helps in absorption of nitrogen. Boron is required for development of new cells in meristematic tissue. Boron is necessary for proper pollination of fruit and seed setting. It helps for formation of nodules in legumes. Boron regulates carbohydrate metabolism. The next micronutrient is molybdenum. Molybdenum is an essential component of the major enzyme nitrate reductase in plants. Molybdenum requirement of plants is influenced by the form of inorganic nitrogen supplied to the plants. Nitrate and ammonia forms effectively lowers its needs. Molybdenum has an essential role in iron absorption and translocation in plants. The next one is chlorine. During photosynthesis, it helps in evolution of oxygen. It is a part of anthocyanin and affects protein synthesis. Chlorine increases turgor pressure. And the last micronutrient for us to study is nickel. Nickel is a component of some plant enzymes, most notably urease, which metabolizes urea nitrogen into usable ammonia within the plant. Without nickel, toxic levels of urea can accumulate within the tissue, forming necrotic lesions on the leaf tip. Once now that we have understood the importance of each macro and micronutrient, Let us look at the deficiency symptoms associated with each of these. Nitrogen deficiency symptoms. As we can see on the screen, for nitrogen deficiency symptoms, plants which have nitrogen for only limited growth may exhibit chlorosis, especially in the older leaves. And in severe cases, the leaves first become yellow and then tan as they die. Some plants, like tomatoes and maize, may exhibit a purplish coloration of the stems. We can see the nitrogen deficiency symptoms in the crops like maize, mango on the screen. To manage nitrogen deficiency, we need to apply Jai Kisan urea, or we need to spray Purna 19 at the rate of 5 grams per liter of water for 3 times at 15 days interval. The second one whose deficiency we study is phosphorus. Phosphorus deficiency symptoms can be seen in corn, paddy, and tomato field. And as we can see in these plants, in case of phosphorus deficiency, plants exhibit stunted growth and leaves are often dark green in color. Oldest leaves become dark brown as they die, and maturity because of phosphorus deficiency may be delayed. To manage phosphorus deficiency, we need to apply phosphorus-based fertilizers like Jai Kisan Samrat, which is applied after planting, and Jai Kisan Super 16. We can also spray Atom 61 at the rate of 6 grams per liter of water for 2 times at 10 days interval. The third is potassium. In case of potassium deficiency, yellowing starts from the tip or margin of lower leaves and extends to the center of the leaf base. Yellowing parts become dead spots. Scorching and burning on the margins of the bottom leaves are most common. We can see these symptoms on the images on the screen. We can see potassium deficiency in banana. We can see the burning of the margins on the bottom leaves. We can see it in paddy as well, wherein we can see the yellowing has started from the tips. And we can see it in maize. Again, the tips have become yellowed. To manage potassium deficiency, we need to apply potash-based fertilizers like Jai Kisan Suraksha, MOP, or we can spray Boon 45 at the rate of 7 grams per liter of water for 3 times at 15 days interval. The next nutrient is calcium. The calcium deficiency symptoms are that terminal bud leaf becomes chlorotic white with base remaining green. 1/3 chlorotic portion of tip hooks downward and becomes brittle. Deficiency causes blossom end rot in tomato and tip hooking in cauliflower. This can be seen on the screen. We can see the blossom end rot in tomato, and we can also see the tip hooking in cauliflower. And we can see the calcium deficiency symptoms in citrus plant. These deficiency symptoms are the other ones that we spoke about just now, which is that the tip 1/3 chlorotic portion of tip hooks downward and becomes brittle. To manage calcium deficiency, we need to spray Calnit 19 at the rate of 5 grams per liter of water for 3 times at 15 days interval. The next nutrient is
magnesium.
The deficiency symptoms of
Magnesium
are intervenial chlorosis of older leaves. Magnesium deficiency leads to leaves becoming mottled, and it also affects chlorophyll formation. Deficiency of magnesium causes sand-drawn disease in tobacco. The mottling of leaves and lack of chlorophyll formation can be seen in the guava plant and the rose plant. And we can see the sand-drawn disease in tobacco on the screen. To manage magnesium deficiency, we need to spray the crops with Mangala Magnesium Sulfate at the rate of 5 grams per liter. The next one that we talk about is sulfur. The symptoms of sulfur deficiency are yellowing of leaves and leaves becoming paler than intervenial portion. Sulfur deficiency also leads to occurrence of downward cupping of leaves in tobacco. This can be seen in tomato plant and maize plant. To manage sulfur deficiency, we need to apply the crops with Mangala zinc sulfate at the rate of 10 kgs per acre, or alternatively, we could spray the crops with sulfur 50 at the rate of 7 grams per liter of water for 2 times at 8-day interval. The next nutrient is zinc. Zinc deficiency causes shortening of internodes with excessive branching, rosetting of small, dark green deformed leaves. Upper leaves, in case of zinc deficiency, will show chlorosis on midrib, veins remain green, and dead spots occur in all parts of leaf. We can see zinc deficiency symptoms in paddy field, in acid lime, and in corn. To manage zinc deficiency, we need to spray the crops with Mangala zinc sulfate at the rate of 10 kgs per acre, or alternatively, we could spray the crops with Mangala zinc sulfate at the rate of 4 grams per liter of water for 2 times at 10 days interval. The next nutrient is iron. The iron deficiency symptoms are young leaves develop chlorosis in the intervenial areas, which may develop into white leaves with necrotic spots. Iron deficiency also leads to the veins remaining green while turning the other leaf portion yellow, and they also tend towards whiteness. We can see iron deficiency in the images on the screen in sugarcane, groundnut, and apple. To manage iron deficiency, we need to spray the crops with ferrous sulfate solution at the rate of 10 grams plus lime salt 1 gram per liter of water for 2 times at 10-day interval. The next one is manganese. Manganese deficiency symptoms are intervenial yellowing of young leaves but not tending towards whiteness. Veins, in case of manganese deficiency, remain green. We can see these deficiency symptoms in soybean, gardenia, and sugarcane. To manage manganese deficiency, we need to spray the crops with manganese sulfate at the rate of 2 grams per liter of water for 2 times at 10 days interval. The next nutrient is copper. Copper deficiency symptoms leads to the young leaves becoming dark green, twisted, and deformed. It also leads to the necrotic spots appearing sometimes and occurrence of marginal leaf burning. These deficiency symptoms can be seen in oil palm, maize, and citrus. To manage copper deficiency, we need to spray the crops with copper sulfate at the rate of 4 grams per liter of water. Next we talk about is boron. In boron deficiency, yellowing or chlorosis starts from the base of terminal bud leaf and extends to the tip, resulting in appearance of a whip-like structure and becoming brownish or blackish brown. This can be seen in mango, cauliflower, and paddy plants. To manage this deficiency, we need to spray the crops with Granubur Natur at the rate of 3 kgs per acre. Alternatively, we could spray the crops with Mangala Boron Plus at the rate of 3 milliliter per liters of water for 2 times at 15 days interval. The next one is molybdenum. The deficiency symptoms are reduced and irregular leaf blade formation, intervenial mottling, and chlorosis around the edges of older leaves. It also leads to the necrotic spots at leaf tips and edges. This can be seen in corn, in cucurbits, and tomato. To take care of this deficiency, we need to apply sodium molybdate at the rate of 400 grams per acre by mixing it with fertilizer. The next nutrient is chlorine. The symptoms of deficiency are younger leaves become chlorotic and plants easily wilt. For wheat plants, they become susceptible to diseases when chlorine is deficient. This can be seen in wheat on the screen and also in the citrus plant. To manage chlorine deficiency, we need to spray the crop with white potash at the rate of 5 grams per liter at 7 days interval. Besides knowing about essential plant nutrients, we also need to understand how their plant availability is influenced by soil reaction. Soil reaction is the measure of hydrogen-ion activity of soil-water system. It indicates whether the soil is acidic, neutral, or alkaline in reaction. Since crop growth suffers both under very low, which is strongly acidic, as well as very high, which is alkaline pH condition, appropriate reclamation measures become essential. If the pH of the soil is between 4.5 to 5.5, then the soil is considered highly acidic. And if the range is between 5.6 to 6.5, then it is considered acidic. When the soil pH is ranging between 6.6 to 7.5, then the soil is considered neutral. Soil is considered alkaline when pH is ranging between 7.6 to 8.5, and when the pH increases to 8.6 to 10.0, then the soil is considered highly alkaline. A pH of 7 is considered best for the plants to absorb most nutrients, as we can see in the chart. When we have a decreasing soil pH from 7, it results in reduced microbial activity, low organic matter decomposition, fixation of phosphates, and it also leads to deficiencies of potassium, calcium, magnesium, and aluminium toxicity. And when the soil pH is above 7, it results in phosphate deficiency. It also results in iron, manganese, and zinc deficiency. It leads to sodium hazard and also leads to reduced water movement. Also, boron availability is very low in acidic and alkaline soils, and as discussed before, most of the nutrients are available to the plants at neutral neutral pH. Hence, knowing pH of the soil not only helps for soil reclamation, but also helps in increasing the fertilizer use efficiency and better macro mesofauna. This concludes our module. Thank you.